Hello, everyone, and welcome to today's live broadcast, Organ on a Chip, Improving Biological Insight Using In Vitro Microtechnology. It is presented by Sabeth Verpoorte, PhD, a professor on the faculty of the Science and Engineering Department at the University of Groningen. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located at the far left of your screen and type your question into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Verpoorte. I will now turn the presentation over to her.
Thank you very much, Judy, for this opportunity to talk to this audience, to you all, about this new technology, OrganonaChip, for improving the way we work cells, the way that we study in vitro outside of the body. I always start my presentation by reminding everyone that actually OrganonaChip technology, which is based on LabRoots technology, is just the latest in a long line of technologies that people have applied to studying cells and tissues over the years. And when I say over the years, I really am talking many decades, really. Human beings have always been interested in understanding how their bodies work, and have always also understood that doing experiments to figure that out, that experimentation can be kind of difficult. The reason is, of course, that we can't just simply open ourselves up and take a look inside and isolate the various processes that are going on inside our bodies. That's ethically, of course, not possible. And so medical researchers have long recognized that you need to take cells and tissue outside of the body and to study it outside of the body in order to better understand processes that are going on inside the body. Claude Bernard was a medical researcher, very well known in his day. He too recognized the need for in vitro research to understand in vivo processes. In his study on experimental medicine, he also noted that physiological occurrence must, as far as possible, be isolated outside the organism by means of experimental procedures. The isolation can then allow us to see and understand better the deepest associations of the phenomena so that their vital role may be followed later in the organism. The interesting phrase in statement is experimental procedures. He was a little vague here. He didn't really specify what kinds of experimental procedures he was talking about. And that's probably because they were very dependent on the type of experiments that he wanted to do. What he and researchers ever since have been confronted with, of course, is how to take tissues and cells out of the human body and to keep them alive, and to keep them outside the human body. The issue is not only just keeping them alive, but also keeping them functional so that they behave and function as they do in the body as well. Researchers have long been confronted by the challenge of developing containers in which cells and tissue can be kept not only alive but also functional in a way that allows the information gained from those in vitro experiments to be extrapolated back to the in vivo situation. So in that context, then, there have been many different ways that people have done in vitro assays based on cells and tissue. What is shown in this particular slide, of course, is a very familiar form for experimentation, the microtiter or microwell plates. These plates are quite simple conceptually. They consist of rows of wells, which can be quite variable in terms of their volumes, depending on how many wells you want to fit into a sort of a standard well plate format. And in fact, you have many variations available, microtiter or microwell plates. Volumes can range from milliliters down to nanoliters, depending on how many wells you want to squeeze into that standard format. Clearly, having so many wells next to each other can be quite an advantage if you want to do many experiments at the same time. In fact, it is possible to add solutions to wells, many or several wells at the same time, using multi-pipetting types of approaches or robotic pipetting approaches. However, arguably, if you grow a layer of cells at the bottom of a well to do some sort of test on those cells, you're presenting the cells a cultural environment which is really not particularly reminiscent of the environment that those cells would encounter within the body. And so oftentimes one needs to be careful then in the interpretation of results that are obtained with cells in this very simple format. There are many assays that have been very well optimized for this format. Doing kind of a de novo experiment, you want to work out a particular cellular mechanism or the mechanism for a particular cellular response. This may not be the format that you want to use. This is where microfluidics or lab-on-a-chip technology then comes in. This is a technology which first really appeared in the late 1980s. You see here images which appeared in 1997 on CNN. And so these images have really become quite iconic, I think, for lab-on-a-chip technology. You see on the left somebody holding a lab chip. You see that these devices are very thin and planar, what they contain then are networks of interconnected microchannels, and those channels are formed in one layer of the device, and then covered by a lid of some sort, which has holes in it often, which allow then solutions to be introduced or removed from the channels. That's what you see on the left-hand side of this particular slide. The channels themselves look very much like the photograph on the right-hand side. In this particular case, we're looking at quite a simple channel network. We see two channels that intersect each other. To fit in or to have those channels filled with different kinds of solutions, and in fact, to manipulate those solutions to form small volumes, which can be injected and separated or analyzed or whatever in some particular way. These channels are quite small. As noted here, they are hair-sized. To give you a better feeling for that, what that means, the average human hair has a diameter of between 75 and 100 micrometers, and so that's kind of the size range that people work in when working with lab-on-a-chip devices. The correspondent in this particular case talked about miniaturization of the chemistry lab to devices which are no larger a dime or a quarter. That miniaturization, of course, is really very appealing, simply just from the point of view of being able to make such small things. However, what's really important is the function, the improved function that we achieve with respect to liquid handling when we use devices with channels in this size range. And the other nice thing is that we use microtechnology to make these devices. These are technologies which were developed or started to be developed in the early 1960s to make electronic circuits. And now we can use the same sorts of technology to make interconnected networks of channels for shuttling around and manipulating various volumes of solution in the regime of nanoliters and even down to picoliters and pentiliters. The fact that we're using this type of so-called photolithographic technology to make our systems means that we can make highly integrated systems. Very intricate and dense networks of microchannels that all interconnect with one another. And you see that here on this particular slide. This is an example from 2002 of a microfluidic device, which was 2.5 by 2.5 centimeters, and which contained which contained reaction chambers to carry out reactions in volumes which are less than a nanoliter. In fact, this 1 by 1 inch device contains 2,056 microvalves in order to control solutions flowing to the reaction chambers. You see an expanded view of one of these reaction chambers on the left-hand side of this slide in the center. It's the green strip in the center. You see then various channels leading to that central reaction chamber. The one closest to the reaction chamber is filled with an orange liquid in this case. The next set of channels leading to the reaction chamber is filled with green liquid and so on. But you need then the valves to control when you deliver what reagent or what liquid to the reaction chamber, and we have 2,056 of those integrated into this device. In fact, 256 reaction chambers are integrated into the system, and each one of these holds less than 1 nanoliter of solution. So the technology allows us to do pretty incredible things with respect to making tiny features and highly intricate and quite complex systems. What's fascinating, though, is what you can actually, or how liquids actually behave themselves in these channels. So let's zoom in a little closer and take a look then again at a very simple microchannel network, 2 channels that are crossing each other as shown in these 4 images. These 4 images are taken from a video which shows the definition, injection, and subsequent separation of 2 analytes in a very small amount of solution. The images were taken over a period of 160 milliseconds, as you see indicated in the lower right-hand corner of each of these images. You see then that the vertical channel, or the channel running vertically through this image, is filled with a solution containing two fluorescently labeled amino acids, amine and glycine, this sample solution is manipulated and pumped so that the intersection of is filled with the sample. The arrows in the images were added to indicate in which direction the various different solutions are being pushed in order to carry out the liquid manipulation. The horizontal channel is not actually visible in this very first image at 0 milliseconds because it's filled with a buffer solution. But you see that the buffer solution is also being pushed towards the intersection to help contain that volume of solution there. Once the sample has been formed at the intersection, that very tiny sample is injected, as is shown in image B, it is then subsequently pushed along the separation channel. We're using electric fields here actually to manipulate solutions, and this allows also to separate the fluorescently labeled arginine and glycine as they are as they have different charge to mass ratios and thus migrate at different speeds in the electric field. What's great here, though, is that you see that these two analytes are actually completely separated over a distance of only 300 micrometers in a time of 160 milliseconds. This is a microchip electrophoresis experiment, in fact. If we were to do the same experiment in a conventional capillary electrophoresis instrument, it would take us certainly 10 minutes at least, if not more. And it would require a capillary having a length of 30 to 100 centimeters or so. And by being able to miniaturize the actual separation system, we are able to reduce the time that this analysis requires down from minutes to just milliseconds. And so what this is truly the power of microfluidic technology. It allows us to manipulate solutions on such a small volume scale that we can actually carry out various processes much, much more rapidly and in an automated fashion. The injected amount of solution here, just to let you all know, is 20 picoliters. This is 100,000 times smaller than the smallest volumes that you can form from a micropipette. So that's generally on the order of 1 microliter or so. So 20 picoliters is 100,000 times smaller. The other interesting thing is that the electric field strength here is 1,000 volts per centimeter. Now, if we were doing this using a conventional instrument, we would require a special high voltage power supply to provide that electric field strength. Here, though, we can actually just use a 12-volt battery to achieve the same electric field strength, because we've managed to shorten the distance over which the separation can take place so dramatically. So microfluidics was shown in the early 1990s to be a spectacular way of improving the performance of various analytical techniques. But it's actually ultimately all about fluid and flow control. And so really started to adopt this technology to be able to carry out other types of work. In fact, we very quickly, towards the late 1990s, saw that people working with cells also wanted to adopt this technology for their own purposes. And that's really because, in fact, microchannels have dimensions which are kind of commensurate with the size with the dimensions of cells. Cells also have dimensions on the order of micrometers. They're generally smaller than the hair-sized channels that I was talking about before. But by introducing cells into these confined spaces, we can in fact grow them or cultivate them in environments which are far more controlled than they would be if we cultured those cells in a well plate. And the ability to be able to control the conditions, to microengineer the conditions around cells by placing them in a confined microchannel volume means that we can also think about microengineering those environments such that they more closely resemble the in vivo microenvironment. And so that is the power of microfluidics then for cell culture. We are able to use the microtechnology to mimic in vivo structures. So a microchannel, for instance, could be used to mimic a blood vessel or other small vessel that one finds in organs or in the body. We can use flow to actually perfuse cells or tissue in microchannels. So that allows us to deliver nutrients to those cells and to remove waste products being produced by those cells, much as the body does as well. We can also use microfluidics to define spatiotemporal gradients, which allow us then to very carefully control the concentrations of different species, chemical species that cells are exposed to. That becomes interesting from the point of view of studying cellular response as a function of molecules and so on. So microfluidics offers then a new in vitro technology which allows us to mimic in vivo conditions more accurately for cell experiments. And so having a route to a more in vivo-like environment should also allow us ultimately to get information from cells which are better able to, or which will better allow us to predict cellular response in vivo. And so since the late 1990s, there have been many different examples of cells being cultured in microfluidic devices. And this is a 3D liver co-culture system, which was published by the Griffith Lab at MIT. In this particular case, we see on the left a plate with 12. Each of those wells, there are 2 wells, and the bottom of the left-hand well in each case contains a porous membrane with many, many pores, having a diameter of 150 micrometers or so. Livers are seeded into this left reservoir, and they then occupy these pores to form 3D liver cell cultures, which in fact start to resemble liver tissue accurately. The nice thing about this particular device is that the right-hand reservoir is used as a reservoir for medium, and in the set of pumps in the base of this plate, which allows solution to be slowly perfused through the porous scaffolds containing all of those micro livers, and that is pumped back and forth between the left reservoir and the right reservoir in order to continuously deliver nutrients and remove waste products from those little, essentially little livers. This device then has been used for toxicology screening and drug profiling and so on, and really starts allowing some very interesting types of studies to be carried out. What's very nice about this type of device too is that it allows us to start thinking about replacing animals with this type of technology so that we can actually reduce animal testing as well. All right, so you can see where this is all headed. I've already used the term micro livers in all of these little pores. In fact, using microtechnologies in general allow us really to start thinking about producing systems with biological models to achieve biological function more on the organ level. So this leads us on this idea of truly replicating organs in the microfluidic devices. But what is an organ on a chip? I think it's always quite useful to be able to distinguish microfluidic device from an actual organ on a chip experiment. And so a very useful definition is one published by Bhatia and Ingber in Nature Biotechnology in 2014. Their definition of an organ on a chip is given here. It's a microfluidic cell culture device created with microchip manufacturing methods that contains continuously perfused chambers inhabited by living cells arranged to simulate tissue and organ level physiology. It's characterized by multicellular architectures, tissue-tissue interfaces, physicochemical microenvironments, and perfusion. And the levels of tissue and organ functionality that are achieved are not possible with conventional 2D or 3D culture systems. So this is an area which is really trending. I will show you probably the seminal example of an organ on a chip, a lung on a chip, in fact, which really got the, people thinking about developing these sorts of systems. It's a paper that was published back in 2010. In November of 2016, I checked to see how many hits I got for organ and a chip in the Web of Science, and it was close to 2,000 papers at that point. I did the same check this month, and in fact, the number of papers now is close to 4,000. So you can see that there's an enormous increase in interest in these types of systems for doing drug testing and so on. But we're seeing that these systems are also being proposed for other emerging application fields as well. All right. The reported organ chip systems in the literature are as diverse as the biological models that they have been designed to study. In fact, most examples that you'll find are based on cell culture models. Models are devices where cells have been introduced into the device and cultured to form some kind of tissue-like construct. I refer to these sorts of examples as bottom-up, comprising bottom-up biological approaches. There are some examples, though, that are based on organotypic tissue samples, where, in fact, actual samples from tissue have been taken and put into the microfluidic device. I refer to these as a top-down biological approach. And in fact, I'll talk a little bit more about work that we've done in this area in a few slides. All right. And so there have been a number of very important examples of cell culture devices which have led to the development, in my view, of this concept of organ on a chip. We see here two early examples. of multi-organ chips based on cell culture. The one on the left, in fact, was first published in 2004. It comes from the Schuler lab at Cornell University. What you see here are is a device containing 4 microfabricated chambers. These researchers introduced cells from representing different organs into each of these individual chambers. So this particular device had a chamber for a lung, for liver, as you can see here, and also for fat cells, as well as an additional chamber for other tissues. These researchers considered an example where naphthalene was metabolized by the liver and then pumped further through the system. It turns out that naphthalene is quite toxic for lung cells, and indeed they showed that cytotoxic effect with their device as a proof of concept. The device on the right is kind of similar. In fact, here we have also 4 different microchannels interconnected with each other, and these channels were filled with cells representing different organs. In this particular case, you see a close-up of one of these compartments. You see that the inner part of the compartment actually contains the cells. That's where the cells are cultured. And it is surrounded by a porous barrier which has been formed by arraying micropillars around the device. And solution is then perfused around the outside of this micropillar array, and nutrients then diffuse into the cell culture area to keep the cells viable and functional. This is really quite nice. It represents a further development with respect to the system on the left. The system on the left simply allowed perfusion of cells directly with solutions flowing over the cell cultures. On the right, the nutrients perfuse into the cell culture from an adjacent channel. This really mimics a little more accurately how that transport happens in the body as well. Okay, but these remain cell cultures. Organ function is not reproduced in these systems. This is an example of a liver on a chip where microtechnology allows also the fabrication of a geometric structure which resembles a liver sinusoid. You see the actual sinusoid on the left-hand side here of this figure, and the microfabricated device at the center. In a sinusoid, liver cells grow next to blood vessels, as is shown here. And the blood vessels are lined with so-called endothelial cells, which regulate transport of compounds from the blood into the liver tissue. That endothelial cell barrier is mimicked in the microfabricated device by an array of channels having very tiny dimensions, and you see that here in the center. Cells then are introduced into the center of the chamber here and cultivated in this way. Here we see the beginnings of an organ on a chip, because the device also incorporates a geometric structure which resembles the actual organ structure, or at least a unit of the liver in the body. This, though, is the device that really gave an enormous impetus or kick-started the whole OrganonaChip development. This was published in 2010 by Daniel He and Don Ingber and Geraldine Hamilton at the Wyss Institute at Harvard. This device mimics the interface between a tiny lung sac or alveolus and the circulatory system. You see then a schematic diagram of one of these tiny lung sacs at the top in the center of this figure. Underneath, you see how a microfabricated device recapitulates this particular interface. The device consists of two halves, or a top half and a bottom half, which when put together forms sealed channels. You see the central chamber is divided into two parts by a porous membrane, which is also microfabricated. Epithelial cells, which are cultured on the top of this membrane, endothelial cells are cultured on the bottom side of this membrane. And the epithelial cells are exposed to air, so they represent the lungs, the side of the barrier exposed to air. The endothelial cells are then exposed to medium in this particular case. All right, so we've got a biological culture containing two different types of cells, which represent this very important interface in the body. However, what's very unique about this device is that it also breathes. The two side chambers, in fact, allow that chip to breathe. The device is made actually in a very flexible silicone rubber material, and so that allows then the channels to be deformed. What you see here at the center of the lower part of this figure is what happens when vacuum is applied to those two side channels. You see that the side channels shrink, and that causes the central membrane with cells to stretch. You can imagine then if you apply vacuum and then release vacuum in an oscillating or an alternating sort of way, that you can stretch and contract the membrane at the center of this device in a regular fashion as well, so to mimic what happens to this interface when we actually breathe. So you see here pictures of the device as well. And so these characterize their lung on a chip. We see here what happens when the central membrane with cells was exposed to silicon nanoparticles with and without stretching. In fact, let's take a look at the photographs in panel A here at the left side. In fact, the intercellular adhesion molecule, ICAM, is upregulated upon exposure to silicon nanoparticles. So certainly the epithelial cells do not like the presence of the silicon nanoparticles. They do respond to the fact that those particles are introduced to that chamber. Interestingly, neutrophils, or white blood cells, are also adhered to the lower endothelial layer when silicon nanoparticles are introduced to the lung part, the air part of the system. And the quantitative gene expression then is given in the figure to the right. You see that for a control device with no silicon nanoparticles added, that the ICAM expression is quite low. That the same is true actually when those cells are being, when the device is actually breathing with 10% strain. However, the introduction of 50 micrograms of nanoparticles per milliliter to the device results in an upregulation of ICAM, even when the device is not breathing. But you see that upregulation, so that response to the nanoparticles increases significantly when the device is actually breathing. And so that's actually kind of results. You see here a different representation of the same data in panel H to the right-hand side here on this slide. We see that when the membrane is in the same, that in fact the percent translocation of nanoparticles through that cell barrier layer is quite low. It's less than 1%. However, when the device is made to breathe, we see a significantly higher percentage of translocation of silicon nanoparticles through the barrier. This, too, is quite a disturbing result because it appears that the application of location of nanoparticles alveolar barrier into the bloodstream, in fact. If we, this type of experiment done in a conventional transwell, these are the white triangles in this particular figure, where also no strain is applied to the cell layer. These conventional systems also do not actually predict this high translocation of nanoparticles. And so it looks, from this type of experiment, it appears that perhaps the conventional format for doing these types of experiments may have underpredicted the effect of nanoparticles in the lung. And so one begins to see that adding this organ function to our micro devices changes the way that the cells respond to external stimuli. Okay, so ultimately what researchers now would like to do, of course, is to combine many different modules or compartments containing different organs in order to be able to study more systemic responses to external stimuli. All right, I'll talk briefly then about an organotypic model for OrganonaChip, a top-down biological model that we've been working with in my lab. We in Groningen have worked over the years with a so-called top-down liver slice model for ADME-tox screening. And this is a model developed by my colleague Henny Groothuis. And it involves taking a very small core of liver tissue from a rat liver or mouse liver generally, also sometimes from human liver tissue if it is available from the hospital. That core, you see that happening in picture A. On picture C, you see the small piece of liver tissue being mounted on a pedestal that is then inserted into a slicing machine, such as shown in panel D. In that slicing machine, a blade then passes very slowly over the top of the core to produce thin slices of liver. What these liver slices look like in panels E and F. As they are sliced, those slices are collected in ice-cold preservation. So what we have here too are ultimately microlivers as well, really, with the added advantage that the architecture of the liver tissue is preserved in these samples, and these samples also contain endogenous enzymes and so on. That's particularly important for metabolic experiments, such as those done for ADME tox screening, and hence this is a model which is very effective for drug development. However, the problem with this model is that these liver slices work very well for 24 hours, or tended to work very well for 24 hours, but work kind of short-lived beyond that. Experiments longer than 24 hours were not possible. And so a number of years ago, my colleague and I decided to see what we could do with microfluidics to perhaps show you how the Rooij lab worked with liver slices. You see here the 12 well plates that liver slices were put into, so one slice per well. The slices are submerged in medium and then placed on a shaker in an incubator at 37 degrees Celsius and in an atmosphere of 95% oxygen and 5% CO2. And then samples are collected periodically after a number of hours from those wells. They're then kept and analyzed separately. And so the medium that the slices are exposed to contain drugs of some sort, and what the samples then contain are the metabolites that are formed when the liver slices metabolize those drugs. Now, you would think that this is quite a sort of a simple way, a simplistic way of doing this type of experiment. People in the microfluidics world would look at this type of thing and say, well, we can do this type of experiment much better under conditions which are more in vivo-like. However, these assays are extremely well optimized with respect to in vivo experiments. And so, in fact, this proved it was quite a challenge for us because we needed to produce a system which was at least as good as this particular assay. All right, so our hypothesis was that perfusion should prolong slice metabolic activity. Here we see a chamber that which can cross section. It consists of two halves, and the inlet is at the bottom. The bottom of the chamber. Medium is introduced there. It passes through a porous polycarbonate membrane. Is pushed upwards through the chamber. When the two halves are brought together, then the medium can be collected at the outlet for further analysis. The idea then, in fact, is to insert or to suspend a liver slice in this chamber and to allow the medium to very slowly perifuse actually around the slice and back out again. And so a compound of some sort in the medium is metabolized by the liver slice, and the metabolites are collected in the solution and analyzed later. This particular device then has a volume of 25 microliters. It's really quite different from the device, from the wells, well plate assay. The flow through the device is 10 microliters per minute. Continuous delivery of nutrients and removal of waste. All right, so the initial experiments involved allowing liver slices to metabolize 7-ethoxycoumarin. 7-ethoxycoumarin, as you see on the right-hand side of this slide, is metabolized in the first phase to 7-hydroxycoumarin. That is accomplished by CYP450 enzymes in the liver. It can be 7-hydroxycoumarin can be further metabolized by other enzymes to glucuronide or several other metabolites. So in an initial set of experiments, we placed liver slices in wells and in our biochip and looked at collected samples from both systems. And we analyzed then those samples for metabolite formation. You see that all three metabolites are formed, in fact, at the same concentrations in both our biochip as well as in the well. The concentrations of metabolites were the same for one liver slice in one chamber and for 2 liver slices in 2 separate chambers, which were linked to one another, with sample being collected at the outlet of the second chamber. So this was a good result because it meant that our device could do at least as well as the well plate. What you see here, though, is that these slices were exposed to a very high concentration of the 7-ethoxycoumarin, quite toxic concentration for the liver. Professor Fotais always used such high concentrations in the wells in order to ensure that there was an excess of this compound present in the well, so that the enzymes, metabolic enzymes, were always working at their maximum turnover rate throughout the course of an experiment. So that the liver slice wasn't actually running out of ethoxycoumarin. However, clearly it would be advantageous to work at much lower concentrations. And in fact, our microfluidic system allows us to do that. Here we see the cumulative metabolite formation over time for a liver slice when the concentration of 7-ethoxycoumarin has been reduced to 75 micromolar. And in fact, we see that the metabolic formation rate is constant over time for all of these metabolites, which means that the nutrients are being delivered at a constant rate and that the enzymes are also functioning at a constant rate. So constant substrate delivery means constant metabolite production. It also means constant collection of metabolites in small volume flow in our system. And that's great because if we can compress our metabolite molecules into smaller volumes, it means we have higher concentrations available and thus better detectability right from the beginning of our experiments. And that's a type of experiment which is not possible in the well, simply because we would be depleting the substrate quite rapidly in the well at these lower concentrations. All right. So our original hypothesis was that microfluidics could yield longer viability and metabolic activity in liver slices. In fact, that hypothesis was not satisfied. We did not see an improvement in the 24-hour limit observed in well plates. However, that's what that told us, in fact, taught us was that microfluidics is not enough to mimic the in vivo situation. However, if you combine flow with other factors, you can provide suitable environments for slices which do allow improved viability. For instance, embedding slices in matrigel improves the phase 2 metabolism to 3 days, in fact. And in fact, selecting the right medium composition also allows slice cultures up to 5 days, as we showed in 2007. Okay, microfluidics enables dynamic control of in vitro experimental conditions, which is a allows us to do interesting experiments, such as real-time online monitoring of drug metabolites or in vitro studies of organ interaction. This is an liver, in fact, where a medium is introduced to the slice in the central chamber here. In that medium, differing levels of an inhibitor for a metabolic enzyme in the liver slice is added. And so one can vary in a controlled way the concentration of inhibitor being delivered to the slice over time, and thus look at the influence of drug-drug interactions on metabolism. Here we see how this experiment was conducted. The concentration of inhibitor, floxine B, was increased by 20 micromolar every 2 hours. And that resulted in the 7-hydroxycoumarin being produced to be slowly but surely decreased over time. You see that here in the graph on the right-hand side of this slide. What's also interesting is this following experiment. In this particular case, the outlet of 3 different chambers or devices was linked to 3 different injection loops for an HPL chromatographic separation. It takes 10 minutes to fill an injection loop, so if you can have a fast enough separation, in fact, also around on the order of 10 minutes or so, one can sequentially fill and inject the contents of each injection loop in a regular manner to get regular information about what's happening in all 3 of those chambers. So to monitor in almost real time the metabolites being produced by slices in those 3 separate chambers. And so that allows for the analysis of metabolites produced by the liver upon exposure to different sorts of drugs. This is, we looked at the analysis of the painkiller diclofenac and its metabolites. You see those 4 metabolites in this particular slide. And this is the metabolite of particular interest here because it's chemically unstable and turns out to be quite reactive actually as well. This is a metabolite which is also nephrotoxic, so toxic for the kidneys. And so it's important to be able to study how much of this metabolite is produced by the liver under differing conditions. However, because it's unstable, you in fact see the concentration of that particular analyte decrease over time. And that then masks actually how much of this metabolite is actually being produced by the liver and what's actually happening to it afterwards. If we do this in our microflow system, we see that we get a far more accurate picture of how much of this particular metabolite is being over time. And you get here then a realistic perspective, in fact, of how much of this particular metabolite is being produced. All right, we can also carry out inter-organ experiments. Here what we've done is we've put an intestinal slice in the left-hand compartment and linked the outlet of this chamber to a second chamber containing a liver slice. Just to look at organ interaction and the regulation of bile acid synthesis by the liver with bile acid. In fact, the liver produces bile acid, the enzyme that does this is the CEP7A1 enzyme. If the liver senses bile acid in its environment, it will tell this enzyme to produce less bile acid or to downregulate that enzyme. Now, of course, the liver in the body is preceded by the intestine, And as it turns, the ileum, part of the intestine, also is affected by the presence of bile acid. It causes, in fact, the formation of fibroblast growth factor 15, which then also has an effect on bile acid synthesis in the liver. It, in fact, also down-regulates that synthesis. Okay, so we have here something being produced at the intestine having an effect at the liver. And in fact, this is an interaction that happens in the body. In fact, we can reproduce that in our microfluidic device as well. On the left-hand side, we see the gene expression for FGF15. We see that, in fact, this gene is upregulated when intestinal slices are exposed to medium containing bile acid. So the slices are behaving as they should. We then take a look at what happens to the liver slices upon exposure to medium containing bile acid. If we look at the single perfused liver slice exposed to CDCA, we see a 25% downregulation of bile acid synthesis. However, look at what happens when we put an intestinal slice in series with the liver slice. We see a further downregulation of bile acid synthesis by 60% actually. Of how, in fact, we can reproduce regulatory processes in the body in our microfluidic devices. And I think this has great potential, in fact, for carrying out not only response types of experiment handle on more fundamental biological processes in the body. All right, so with the OrganonaChip that bridge the in vitro in gap, we can think about integrating analytical function into our system. For the experiments that I showed you up until now, was done offline generally, although sometimes we link our microfluidic devices to directly into our microsystem for controlling incubation conditions, oxygen, for instance, but also for analyzing cell metabolic products. And so think about placing sensors at the inlets and outlets of various compartments of our intestinal liver system as well, for instance. Now, the determination of oxygen is kind of can be kind of tricky if you want to miniaturize it, because even small sensors can take up a lot of space in our organ. Example in this particular photograph. You see the discs and the liver slices, but you see what happens when you stick even smaller optical fibers into these 25 micrometer chips. If you get a bit unlucky, in fact, you end up pushing the liver slices aside, or otherwise perhaps damaging them even by inserting your sensors. We've then come up with nicely designed liver slice perfusion systems in which we can actually determine oxygen consumption of the slice. In this case, we see the slice chamber in the center. We place an oxygen sensor before and after that chamber in order to measure consumption of oxygen by the liver slice in the chamber itself. And you see here then pictures of how we managed to accomplish that. And for slice, we see an oxygen concentration of around 600 micromolar. That oxygen concentration is the same as for the situation where we have where the slice in the chamber is dead. There's no slice in the chamber. You see a significantly smaller amount of measured oxygen at the outlet of the device, meaning that indeed this is consuming oxygen as we would expect. All right, to conclude then, a biological model, organ chips must be environment for cells. The need for transport to and through an organ chip system, but that allows for integration compartments into multi-organ systems and for online analysis, so we can use it to our advantage. Microtechnology is great because it allows us to make microfluidic devices, but it also makes features in these devices which allow for mechanical stimulation of cells and integrated sensing as well. Application areas include drug development, but also personalized development and medicine, especially when patient stem cells are being used. So up and coming applications then are for disease modeling and regenerative medicine. With that, I end my presentation. I just want to acknowledge my group member been involved with this work over the years. Paul van Nitwoud, the smiling young man at the lower left, did a lot of the work, initial work for developing our chambers for liver slice. Guy second from the left in the photograph in the top row above, Peter Oman, integrated oxygen sensors into our systems, and I have had other people work on cells and so on. Thank you for your attention, and I'd be interested to hear what thoughts you have.
Thank you, Dr. Verpoorte, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window and type your question into the box that appears on your screen. We'll answer as many of your questions as we have time for, so let's get started. Our first question is, given the definition you gave for organ on a chip, could either of these two examples be considered organ-on-a-chip systems?
Okay, so I guess you're probably referring to these multi-organ devices that I showed early on in the presentation, where sensors were being used to represent the different organs. And in fact, I've really started to answer the question actually by giving the description of those two separate devices. The fact that cell cultures were being used means that we're not quite at the level of organ function in these devices, which is representative of an organ. A cell culture in these devices anyway consisted of single layers of cells. A single layer of cells does not really represent tissue and does not really allow the recapitulation of function on a physiological function on an organ level. So those initial devices from 2004 and slightly later were very important examples at the time. They really stimulated people to start thinking about how to recreate organ function in devices. But those devices themselves did not allow that organ function just yet.
Thank you. The next one is, what materials could be used to make the chips that house the cells or tissues which comprise organs on a chip? And what are the typical dimensions for these systems?
So many different sorts of materials are available for making microfluidic devices these days. Traditional material, glass is a nice material because it's transparent and we can look into. So we can place a glass device under a microscope and actually look into it, and thus monitor cell behavior and so on. And I think that's an important aspect to mention here. A lot of the functional studies for organ-on-a-chip devices are done using microscopes, and so it becomes important to use a transparent material of some kind. Glass is one such material, as I've already mentioned. However, people are generally turning to polymers of some sort to make these organ-on-a-chip devices. Silicone rubber material has been used, and that's an interesting material if you want to or whatever. But it's also possible to use materials which have been used to make well plates, and so which we know are biocompatible. I'm thinking of materials like polystyrene or polycarbonate or whatever. The chambers in these devices tend to be a little bigger than the microchannels I showed you at the very beginning of the talk when I was introducing microfluidics technology for doing separations of amino acids and that sort of thing. We can make very small channels on the order of the diameter of a hair or even smaller. However, if you want to recreate organ function, you need larger numbers of cells. You need generally thousands of cells. And so slightly larger chambers then, which have volumes more on the order of a few micrometers, and so on. But yeah, again, it depends very much on the application and the type of cell experiment that you'd like to carry out.
Thank you. We have time for one more question. Could the interval between measurements in this example be shortened in some way to capture possible processes occurring on a much shorter time scale?
So indeed, I think here we're referring to the analytical results that I showed for the metabolism by liver slices in microfluidic systems of substrates like 7-ethoxycoumarin. In those studies, we were sampling medium from the perfused liver slice incubation chambers. That sampling was happening once every half hour or hour, and analysis information about liver slice metabolism every half hour or hour. In fact, we could get information far more regularly if we went to an approach where we sampled much smaller volumes of solution and perhaps did separations in microchannels as small as the ones that I showed at the very beginning of this presentation. So if we could sample just a few liters at a time from our incubation device and directly inject then those samples into microfluidic separation devices, we could be getting information far more regularly, on the order of every minute even. And that could give us a bit more insight as to whether there were processes happening on a much shorter time scale.
Thank you. I would like to once again thank Dr. Verpoorte for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 31st, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.